Hey guys, this is Nick. I noticed my last video was a little long, so just decided to do a different one because my voice was really messed up last time, so now I'm a little bit better. Alright, this is genome sequencing, the future of personalized medicine. What is genome sequencing? Genome sequencing is figuring out the orders of DNA nucleotides or bases in a genome, the orders of A's, C's, G's, and T's that make up the organism's DNA. The human genome is made up of over 3 billion of these genetic letters. Alfred Storpent created the first Drosophila gene map in 1911. Drosophila was a small fruit fly used extensively in genetic research because of its large chromosomes, numerous varieties, and rapid rate of reproduction. The crucial first step in the molecular genome analysis and in the molecular biological research of the last half century was the discovery of the double helical structure of the DNA molecule in 1953 by Francis Crick and James Watson. In the mid-1970s, Frederick Sanger developed techniques to sequence DNA. The United States Department of Energy, seeking the data to protect the genome from mutagenic gene mutating effects of radiation, became involved in 1986 and established an early genome project in 1987. James Watson was appointed the lead of the NIH component, which dubbed the Office of Human Genome Research. The following year, the Office of Human Genome Research evolved into the National Center of Human Genome Research. The advent and employment of improved research techniques includes the use and restriction of fragment-length polymorphisms and polymerase chain reactions, bacterial and yeast artificial chromosomes, and pulsed gel field electrophoresis. These are all methods of just finding your DNA sequence. Enabled rapid early progress. Therefore, the 1990 plan was updated with a new 5-year plan announced in 1993 in the journal Science. In 1993, the NCHGR established the Division of Intramural Research (DIR), in which the genome technology is developed and used to study specific diseases. By 1996, 8 NIH institutes and centers had been collaborated to create the Center of Inherited Disease and Research (CIDR) for the study of genetic and complex diseases. In June 2000 came the announcement that the majority of the human genome had in fact been sequenced, which was followed by a publication of 90% of the sequence of the genome's 3 billion base pairs in the journal Nature in February 2001. Surprises accompanying the sequence publication included that relatively small numbers of human genomes, perhaps as few as 30,000, um, of the complex architecture of human proteins compared to their homologs. Similar genes with the same functions in, for example, roundworms and fruit flies, and the lessons to be taught by repeat sequences of DNA. What is personalized medicine? Many doctors will tell you that they have been practicing personalized medicine for years, and that would be true if they used Wikipedia's definition of personalized medicine, but the definition preferred by the National Human Genome Research Institute is more specific, maintaining that the personalized approach to medicine includes an individual's genetic profile to guide decisions made up in regard to the prevention, diagnosis, and treatment of the disease. Oh, oops. And here's just like a little diagram for all of you to see what goes on. Alright, the advantages, the benefits to personalized medicine and genome sequencing. Advantages. Creating personalized plans to treat disease may be possible based not only on mutant genes causing the disease, but also on other genes in the patient genome. Genotyping cancer cells and understanding what genes are misregulated allows physicians to select the best chemotherapy and potentially expose the patient to the less toxic treatment since the therapy is tailored. Previously unknown genes may be identified contributing to disease state. Traditional genetic testing looks only at the common troublemaker genes. Lifestyle and environmental changes can mediate the effects of genetic predisposition but may be identified and then moderated. Disadvantages. The role of most genes in the human genome are still unknown and completely misunderstood. Therefore, a lot of information found in the human genome sequence is unusable at present. Most physicians are not trained on how to interpret genomic data. An individual's genome may contain information that they do not want to know. For example, a patient has genome sequencing performed to determine the most effective treatment plan for high cholesterol. In the process, researchers discover an unrelated allele that assures the terminal disease with no effective treatment. The volume the volume of information contained in the genome sequence is vast, and policies and security measures to maintain the privacy of safety of this information is still new, because people don't want their, genome sequencing, their little literal genetic makeup out on the market. The future of genome sequencing. Since the completion of the Human Genome Project, technological improvements in automation have made increased speed, lower cost points, and individual genes can be sequenced routinely, and labs can sequence well over 100 billion base pairs per year, and an entire genome can be sequenced in just a few $1,000s. Many of these new technologies were developed with the support of the National Human Genome Research Institute, General Technology Programs and its Advanced DNA Sequencing Technology Awards. One of the NHGIR's goals is to promote new technology that could eventually reduce the cost of sequencing a human genome or even higher quality than is possible today for less than $1,000. One new technology involving watching DNA polymerase molecules and their copy their DNA. The same molecule can be made up of new copies of DNA in the cells with a very fast movie camera and microscope, and incorporating different colors of bright dyes, each for a different letter A, T, C, and G. This method provides different and very valuable information than what's provided by the systems that are most used today. Another new technology um, in development entails the use of nanopores to sequence DNA. Nanopores based DNA sequencing involves threading singular DNA strands through an extremely tiny pores in a membrane. DNA bases can be read one at a time as they squeeze through the nanopore. The bases are identified measuring the differences in their effect on the ions and electrical current flowing through the pore. Using nanopores to sequence DNA often offers many potential advantages to the current methods. Is for sequencing to cost less and be done faster. Unlike sequencing methods currently in use, nanopore DNA sequencing means researchers can study the same molecule over and over again. Alright, why should you care? The genetic sequencing is a predominant tool used in personalized or precision medicine, which monitors people's DNAs and biomarkers to improve their health care. Precision medicine allows doctors and healthy patients to their disease risk and match sick patients and effective treatment plans. This could mean the end to all genetic diseases, or at least a much faster way of diagnosing someone before they have even developed the disease or show any symptoms, meaning patients could use alternative methods to cure these diseases before the even riskier ones were even an option. And that is the end of my presentation. I hope you all enjoyed, and thank you for coming. Goodbye.